Hello guys, welcome back to my videos. In today's lecture, we are going to see about the Griffith experiment of transformation. Now, what is meant by transformation? Transformation is nothing but the process or vital process present in the bacterial cell or bacterial organisms in which exogenous genetic material is transferred from one cell to another cell through cell membranes under competent state. That means in favorable conditions, genetic material is transferred from one cell to another cell through cell membrane is called transformation. This is nothing but the genetic process or genetic material exchange process. Even though in earlier histories or in earlier experiments done by scientists were doing lot of experiments but they were unaware that genetic material or DNA is a sort of genetic material present in the microorganism. So earlier protein was considered as a genetic material which transferred from one generation to another. But later in 1928, Frederick Griffith conducted the experiment which conclude that nucleic acids in the form of transforming principles act as a genetic material. Now, what type of strains or organisms used during the experiments by Griffith? The strain used was Streptococcus pneumoniae or Pneumococcus pneumoniae, and lab animal were used was mice. So during the experiment of conducting the experiment of pneumonia disease, Griffith found some different factors. Let's see. So Streptococcus pneumoniae or Pneumococcus pneumoniae having two types of strains, that means strain S, which is highly virulent and infectious, while as strain 2 was non-virulent, that means not infectious as much as strain S. So strain S was having smooth colonies, that means they produce smooth colonies on agar, and strain R were producing rough colonies on agar. And strain S was capsulated as shown in the figure, and strain R was non-capsulated, that means they are non-virulent, thus they do not produce capsules over their cell membrane. Because of the capsule, strain S were pathogenic in nature, and thus capsule was not present in strain R, they were non-pathogenic in nature. Thus strain S were causing disease, that is pneumonia, but strain R is non-virulent, non-pathogenic, non-infectious, that thus does not cause any type of disease. And there are four different types of strains are there. S has 1 2 3 4, R has 1 2 3 4. Now coming to the experiment part, Griffith conducted the experiment using Streptococcus pneumoniae and mice. So first he used strain 3 organism and he inject it into the mice. When mice dies, he isolated strain 3 because it was capsulated organism and infectious, thus it caused the disease pneumonia in mice, and due to that mice gets die. Then in second turn he used R2 type of strain when it was injected into the mice, because it is non-virulent, non-pathogenic, non-capsulated. Mice does not die because it does not get disease, thus it gets alive. And what did he isolated from that live organism? R2 strain. In the third turn he killed strain 3 of pneumococci via heat, high temperature. Using high temperature he killed strain 3 microorganism and he injected into the mice. What he found that mice became alive, or as a result mice was not die because the capsule was denaturated due to high heat. Thus he isolated heat killed strain 3. But in the fourth turn he obtained an amazing result, as when he used heat killed strain 3 microorganism, and combined with R2 microorganism, R2 strain you can say, and injected into the mice. What he found that mice gets die. Why? Because there he conducted or he concluded that there is something present in heat killed S3 strain which was transferred from heat killed S3 to R2. Thus R2 becomes virulent, and thus he obtained in the result or in died mouse as S2 strain. So this was the experiment conducted by Griffith, and in the fourth turn he found out there was a something factor or the thing which was present in heat killed strain 3 which was transferred from HKS, that means heat killed S3 to R2, and becoming R2 strain as a virulent S3 strain, and thus the transforming factor of Griffith was termed as transforming principle. So this was the experiment conducted by Griffith to conclude there is something present as a genetic material which is in the form of transforming principle. Thanks for watching.